us? Yeah, she's here, but she's not answering. So maybe she's getting also a coffee.
Yes, I am.
Okay, so you can hear me, you can see me, I can hear you, I can see you. Wonderful.
Yeah, it's working.
Okay. And can you also confirm that are we sharing the screen? No.
I thought you were going to share the screen because
Yes, I will share my screen and then I will bring up your presentation. But before we do that, I say just a few words about the session. Yeah. So Ellen, maybe give me 2 minutes or maybe 1 minute. Okay, can we close the door? I hope you all have enough oxygen in the room. Very quickly, my name is Christian Breuer. Some of you know me, some of you don't know me. At least the people who are doing the SusCrop second call projects know me because I sent them the letters. I used to coordinate the entire SusCrop action, which was an AeroNet co-fund action, which is officially finished now. Final payment from the commission is coming soon, so that's very good. But still, a second call is running, a third call is running, and a fourth call is the agro-biodiversity call is also still running these projects. So, but anyways, that's what the Green Arrow Hub is doing now with Facho JPI. We're taking care of these projects, allow these kind of status seminars. Yes, so and I think with this, I think we can start the session. Luckily, you have decided to choose the best session of the parallel sessions. So, Ellen, I give the word to you, and Ellen is gonna present the Roots Plus project of the second call.
I don't see the presentation yet, so.
You don't see the presentation?
No.
Oh.
I see a running circle.
So I'll try that again. Can you see your slides now?
No.
No? Okay, well, we can hear you. Can you open your presentation? I mean, the presentation on your laptop,
Yeah,
and then
I'm checking.
And then I try to synchronize while you're talking. You just have to tell me when you shift the slides.
But do you want me to share my presentation? Because I don't know if the other people online see it.
Yeah, that might be good. Let's try this. Yeah, that's actually much better. Yeah. Okay. Let's see, where are you? Can you share them already, or do I need to make you?
I'm checking how to do it.
Okay, let's try. Let's try. What's this here? Yeah, that looks good.
Okay.
Yes, can you go to presentation mode?
It should be in presentation mode, but maybe this is because I have two screens. Wait. It's better like this or not?
No. I think we can't see the screen with the presentation. I mean, we see the PowerPoint, but not in presentation.
I'm gonna. And now?
Yeah, we always need to wait 2 or 3 seconds.
You have to share again. I'm not sharing it.
Do you see it in presentation mode now, or?
No, we don't see anything. You're not sharing at the moment. You're sharing them.
Um,
Yeah, no, we see it, but it's not in the presentation.
Wait, I'm trying some things.
Yeah.
Is it okay now? I don't think you see it in presentation mode.
No, we don't see it in presentation mode, but that's
I
right.
don't know how to solve it. I'm not familiar with this WebEx thing, so can we do it in this way?
We can also do it this way. I open your presentation for the people here. And when you shift the slide, just tell me, then I also shift the slide here.
Okay, I'll try
Okay.
to make this larger. Okay, so today I want to talk about the results of Roots Plus, a project that was finished the end of March. Yeah, now I have troubles. Next slide. So the objectives of the project, well, we had 7 in total, but maybe first I explain briefly how the project was running. Does everyone see my second slide? Because I have different screens and I don't know anymore.
No, but you're right.
Okay. So what we actually did is we co-cultivated a bacterial strain, Rhizobium rhizogenus, with some plant material, tissue culture material. After this co-cultivation, this resulted in the formation of hairy roots, what you see on the right-hand side. And if we were very, very lucky, out of these hairy roots, new shoots would be appearing, and we had RI plants. These RI plants contain the a part of the DNA that was transferred from the bacteria to the plant, meaning a part of the RI plasmid, what you see on top right of the slides. And this RI plasmid contains the tDNA. It's the tDNA that's being transferred, and on this tDNA there are the typical role genes, roots, oncogenic loci, and that are the genes that are also responsible for the hairy root phenotype that you can see on the picture on the right-hand side. So I will not go through all the objectives here, but we will immediately go to the results. Okay, first I will briefly explain our partners. So this project was coordinated by ILVO on the Belgian side, and we had 2 private breeding companies that were willing to share their material and their expertise with us. From Germany, we had a group of Professor Traut Winkelmann and Professor Debener. In Poland, we had people from the Nicolaus Copernicus University in Torun, and in Romania, we had a partner of the University of Agricultural Sciences and Veterinary Medicine in Cluj-Napoca. We were working on 4 model crops, chrysanthemum and apple. That were the crops we were working with in Flanders because of the breeding companies involved. In Germany, they worked with apple and rose. And in Romania, the model crop was sunflower. And the people from Poland actually worked with all the different crops because they were experts in plant physiology. So our first good achievement is that we were able to implement the RI technology in the 4 different model crops we were working on. So
Ellen, tell me when you want the next slide.
Sorry, I'm on the slide. RI technology developed for 4 crops.
Yeah, very good.
Yeah. So you see some pictures here of these small RI plants in tissue culture. For sunflower, however, it was not possible yet to achieve a complete RI plant. Here we had to work with the composite system, what you see on the bottom left. And the sunflower plants that we were able to grow have RI roots, but the rest of the plant is still not containing the genes transferred from the bacteria. Next slide. The regeneration of the plants from the roots remains the biggest bottleneck. It was already a bottleneck at the beginning of the project, but we hoped to be able to find some reasons why regeneration is so difficult and find a clue to solve this problem. One of the theories was that the genes that were transferred, so for the agropine type strains, it's what you see on the top left, the agropine type bacterial strains transfer TL and TR DNA, and on the TR DNA there are actually auxin genes. So we thought, okay, maybe these auxin genes are interfering with the regeneration. We will look if we see differences between roots containing these auxin genes and roots without these auxin genes, but there's no logical correlation between this. We have been checking the addition of cytokinins, auxin transport inhibitors. We have been measuring the hormone balances in some of the roots. And in some cases, we see an upregulation of salicylic acid and jasmonic acid, but it's not that it's in all species we work with that we see this upregulation. In some cases, and that's what you see on the top right, we had spontaneous regeneration while the heavy roots were still on the X plant. So yeah, we were very lucky to have this. This was in chrysanthemum and in rose. But the main take-home message is that the genotype is actually the biggest factor influencing the regeneration. So it's no matter what recipe we use, it will always be species dependent, but for some, in each species, it is also genotype dependent, and some genotypes will never regenerate and some genotypes easily regenerate compared to the others. Next slide. So we grew up some of these RI plants and we compared them to wild type plants. This is an example in rose, where you see on the left-hand side a control plant without the RI genes, and then you have 3 plants with only the TL DNA genes, and then 3 plants with both the TL and the TR DNA genes. You also see here that there is no logical correlation between the phenotype and the presence of this TR DNA fragment. You see that there is clearly compactness present, but it's not always the case. On the left bottom, you see that we have a serious reduction in the leaf size. On the bottom in the middle, you see a graph representing the internode length, and also there you have a huge variation in internode length between the different RI plants that were generated. And some of them really are much shorter, have shorter internodes compared to the wild type plant that's marked in blue on the left. And then the picture on the right is showing the rooting of cuttings, and you clearly see that these RI plants have a more intense rooting system compared to the control plants. Next. Then when we were looking in chrysanthemum, we also measured the plant's water household system. And actually, we did not see big differences between the wild type plants and the RI plants. Under normal conditions, they acted more or less the same when we made a comparison. However, when we placed the plants under stress, under drought stress in this case, we did see a difference in response of the RI plants. So on the left-hand side, you see on the top the control plants that were well watered all the time. And on the bottom, you have the plants that received a drought treatment, meaning that they were not watered for 7 days. And always on the left-hand side of the picture, you have the RI line, and on the right-hand side, you have the wild type plant. And when you look at the bottom, you clearly see that the RI line that's on the left is looking better compared to the wild type plant after the drought treatment. You can also see this in the graphs, so the relative water content of the array line, that's the dark green bar, is more or less the same as the control plants that were continuously watered, while the wild type plant that suffered from drought clearly has a lower relative water content, and also the photosynthetic rate is much more upregulated in the array line compared to the wild type. Next slide. I already told it in the pitch. We also looked at the response to apple replant disease and rose replant disease. So here we have a control apple plant and 2 array lines. You clearly see that there is a difference in the phenotype. You also see the rooting. So the rooting system is not always denser compared to the wild type. And these some cuttings of these plants were placed in gamma irradiated soil compared to apple replant diseased soil. And there is a small indication for the array line 2 that the plants don't seem to suffer from the apple replant disease, but for sure more research is needed to be able to say that the RI genes really help plants in withstanding this disease. Next slide. Then we got the most stunning result, and what was also expected because it was already reported in, for instance, Kalangoe, is that the plants were more compact, and for ornamentals this is an essential criteria. A lot of ornamental plants are treated with plant growth regulators to keep them compact or in a certain shape, and we were now able to have plants that can be as compact as a plant growth regulated treated plant. So if you look on the graphs, you always have the curves on the right hand side of the array line 144. And as you can see, the blue bars are without plant growth regulators, the orange ones are with plant growth regulators. And actually this array line 144, of which you also see the pictures on the bottom, is without plant growth regulators of the same diameter and height as the wild type plant with plant growth regulators. So this plant can be grown without the use of chemical treatments. Next slide. Then we also performed, our German partner also performed a GWAS study in rose. So 104 rose genotypes were on the one hand genotyped on the SNP array. The phenotype of callus formation and hairy root formation was determined. All this information was combined in this Manhattan plot, and in this way we could find some positions, some alleles that in a certain when present in a certain dosage had an effect on the hairy root formation and the callus formation of these rose plants. They are now looking at the candidate genes that can be responsible for these correlations. They will be looking for genes being reported in the past involved in transformation or adventitious rooting, and also at difference at gene expression data. Next slide. Then regarding the breeding, this was, since we were working with 2 Belgian breeding companies, this was quite an important aspect of the project, because a company is only interested in applying a technology if they can do something with it in the end. Yeah, what you see on the bottom left is that this is the F2 population, an F2 breeding population of chrysanthemum made from an initial array line. You see a lot of variation in size. We had these 5 initial array lines that were characterized very profoundly. We genotyped these lines with FISH technology to determine at how many positions in the genome the array genes were being transferred. We also determined for the most crucial genes of the TL and the rDNA the copy number of these genes. And these lines were then further selected for breeding. And what you see on the bottom right is a table indicating the genes that are present in the F2 population. And as you can see, we have clear segregation of the genes in the breeding population. We have plants that only contain, for instance, the role BTR gene, that from the LDNA only have role A and role B, and role C and D are gone throughout the breeding. So this makes it very interesting because you can create a lot of variation after using this material in the breeding. Next slide. So what will the future bring? For Apple, we can say that we hope that we will be able to create new rootstock material in this way. At least the breeding company is very interested, the Belgian breeding company, and also in Germany, there are people very interested in these results. The RI plants are now further tested at the breeding company to evaluate how they react in normal, in their facilities. Also for the chrysanthemum, the F1 and F2 plants will be phenotyped thoroughly by the breeding company, and they will also continue breeding with it. For sunflower, phenotyping will be done, and then we have quite some private breeding companies interested in using the technology, and we also have follow-up research projects with multiple partners, for instance, Marie Curie Fellowship, PhD. I find the name. Something with Marie Curie with different partners, where we also hope to continue working on the technology on a more fundamental basis. So this was very briefly what we have been doing the last 3 years. Yeah, if there are any questions, I'm willing to answer them.
Very good. Thank you very much. Now the problem is, I think the people online couldn't see your presentation, I assume. But
I don't know. I tried to share it, but yeah, I didn't know it. They were not seeing it. Otherwise, I would have stopped.
Yeah.
I could only see the presentation, not the chat. So.
Okay, good. Well, or not good, but we can't change it for the moment. So, is there any question in the room? I have a question in the room. You might have to speak up loud.
Yeah, I have two actually. If I look at water use efficiency, and the second one.
I don't understand it. Sorry.
Water use efficiency. Did you look at water use efficiency?
Did you look at water use efficiency?
Yes, it was all measured by the Polish partner. Yes.
Is that what it improved or not? And the second question, nitrogen uptake, cycling, did that improve?
So
sorry,
with the water use efficiency, the question was: the water use efficiency improved?
I should check. Okay. I don't have the full results with me now.
Okay. Second question was
nitrogen uptake. Nitrogen cycling.
No, nitrogen was not checked.
Okay, nitrogen was not checked. Okay. Okay, Peter.
My name is Peter Gregory. I'm not a plant breeder, but is this actually a breeding technology, or is it just a system for producing mutants? Because I didn't see anywhere where you had either a stable genotype or a stable phenotype.
It is a breeding technology, especially interesting for vegetatively propagated crops, because if you have a new genotype that's correlated to a specific phenotype, it's fixed because you vegetatively propagate it, and that makes it very interesting that if you have if you have for instance a nice plant but you want it to be more compact, that you can use this technology to have more or less the same plant but more compact without having to do a lot of breeding steps. So that in that way it should fasten the breeding cycle.
Okay, so it's a way of producing much more variation, which you then
Yes, it is.
use.
Yeah. Of course, you can also do it with some mutation technologies, but yeah, this is more direct, focusing on the rooting and on the compactness.
Okay, my second question is, in your presentation, you talked about RI genes.
Mhm.
Are those unique, and are those, what are they? What are those RI genes?
The RI genes are the genes that are present on the RI plasmid in Rhizobium rhizogenus. So yeah, I showed it, but yeah, I cannot show it on the presentation. It was on one of the first slides. On the top, you had a plasmid, and the RI genes are actually the genes that are present on that plasmid, and in our case, only the ones that are present on the tDNA of that plasmid.
And all of those are transferred into the new plant.
Not necessarily all of them are being transferred. That's why we always determine which genes are being transferred and in what copy numbers and so on. So it can also be that part of the DNA is cut off and that only half of it is transferred.
Thank you.
I think you have to go on time-wise.
Yes, we will continue, but I had to dedicate a little bit more time to the online people because the people here will have the chance to ask questions to the people actually physically in Berlin. So the next speaker is Christian Hermans from the Free University in Brussels with the project C for Future, so C4 plants. Let's see. Actually, I can try to do this, but I think it's not working.
So you have two questions for the price of one.
Session 1. Here we go.
Thank you. So I thank people also for following online. I've seen some familiar face. So the project ended in April, but we prolonged it to the end of the year. So there are still people being hired on the project. So
We are still seeing online the presentation.
they don't see?
It's because she's sharing her presentation. Yes. Maybe Ellen, can you stop sharing?
But we cannot share ours.
I'm not sharing, sorry.
Let me try to share. Maybe.
Yeah, maybe try from this one.
Yeah.
You know that there's the whole presentation.
Yes, I have presentation here. Can the online people now see the roots? Oh, that's the wrong one. Sorry.
Online, we only see a running circle. I think the presentation is too heavy to be shared or something.
You only see a running circle.
Yeah. And a black screen.
Okay. We cannot share any.
No, we cannot share it in this room. We had the problem this morning as well. Yeah.
So you will have the sound, not the image.
Yes.
So you have to image that plants are phototrophic organisms, so they use light as a source of energy to fix CO2 into sugar. That's the basic. And in that task, they require a certain number of soil resources. We are focusing on water and nitrogen. So water limitation, drought is already a major challenge for crop production, and nitrogen is the element required in the greatest amount. It is the plant fuel, but fertilization has a negative impact on the environment. So the project was focusing on resource use efficiency. So this is the biomass you can produce out of 1 unit of these resources. and with CO2 level increasing in the atmosphere, the Earth will favor plants with a C4 metabolism, as I said earlier in the pitch. So these plants, they have an anatomical feature to concentrate CO2, so they are better, more efficient at photosynthesizing, producing biomass, and they are also preventing water loss. So in the consortium, so we're working on identifying some key information to breed crops for traits relevant to climate change. And the two crops we are studying are maize and sorghum. So maize is ranking first for the global cereal production, and sorghum fifth. And in Europe, it is still considered as a niche crop, but a promising one, and it's meeting gluten-free needs compared to maize. So the scientific strategy is exploiting the natural variation in large diversity sets we have assembled for maize and sorghum to identify favorable plant characteristics and genes regulating or markers regulating the resource use efficiency. So we in the project have a special focus on the roots. A larger, deeper branch root system will increase acquisition of these mobile resources, so they're migrating as the season is running. So the most immediate benefit of a deep root system is to access water during prolonged drought episodes. So here, and at the shoot level, maintaining leaf growth would sustain photosynthesis activity during moderate water deficit, and decreasing the leaf area could avoid severe terminal stress. So first, for the laboratory activities, we used a very simple growing system on germination paper like this here. And we can observe the 2D root morphology of sorghum. This is a simple root system, a primary root and several lateral roots. Maize is a little bit more complex, so it has a primary roots, lateral roots, and it has also seminal roots. So these are parallel to the growing parallelly to the primary roots. So after that initial screen, so we selected some genotypes with contrasting root morphology, and we grew them in an automated belt conveyor here with X-ray source in orange, and this is the box getting the image. So this is the same technology being used in hospital to get differences in water densities between tissues, so we can have access to the root system. And this is an example of some contrasting genotypes for sorghum in well water with nitrogen condition, and here you have the nitrogen stress and water nitrogen stress and drought stress combined. So some genotypes they explore essentially the horizon, like this one. So this is a good for the water at the beginning of the season. We have also different genotypes with very steep angle going deep in the soil and not exploring the horizon. And some they have intermediate angle here to maximize the volume of soil. So for the phenotyping of plants in the field, so we have three trial sites in Italy, France, and Turkey, so in the Mediterranean, and also one site in India. This is because one of our partners was transferred to ICRISAT, but we continue to do the trials over there. Yield traits were measured, high throughput field phenomics, and we use also drones. This is the case like the diaphragm platform in south of France in Mogio. You see here the plots with the different maize genotypes being grown all together in control and stress conditions. So don't be afraid of this graphic. It is called a spider plot, like a spider web. I'm representing here all the traits being measured in the field here, so on the traits, and the data are normalized. So the lines here are representing the 280 different genotypes we had in the field, and the blue circle shows 0% variation compared to the mean panel. So one genotype being just here on the circle shows no variation compared to the average of the panel. An example here of large variation is the antithesis silking interval. As you may know, males are female flowers and male flowers, and this has to be synchronized. Maturity has to be synchronized to have pollination. And if we look at the responsiveness, so this is the stress minus the control divided by the control. So this parameter, so ASI, is really popping up. So this is a zoom. And this is the reason why we have a strong impact on the yield, because the reproductive stage is sensitive, so you don't synchronize the maturity of the flowers. Imaging ears was also used. I will not go through very detailed analysis, but this is the minimum and maximum of grain number you can have. So up to 500 in control condition, and in drought and minus nitrogen condition, only 20 grains you will have on the ears. For sorghum, so the trials were carried in Turkey, Adana, so they are preparing the plots here. Aerial view of the plots, control conditions, minus nitrogen and water combined. So the sorghum can reach quite high. The height can be high. You can see the impact of the stress on the panicles with the grain. There's a mistake here. This is control. Then minus nitrogen, and minus water combined. Again, despite the plots to show you the variation we have in the responsiveness to minus nitrogen or minus nitrogen and water combined. And as for maize yield, so there is a strong delay in the physiological maturity days to flowering, and then the panicles also, they mostly contain less grain, but still you can see some lines they pop out, so they apparently they prefer to be under stress condition to produce, and it's quite interesting with sorghum. So some of the genotypes are stimulated by the drought condition. We also analyzed the quality of the grains. In the spider plot here, you see all the nutrients or the mineral elements being measured. For example, here, so there is less magnesium, less zinc here, but in both conditions, the total phenolic content is popping up, so it's increasing, showing that the antioxidant properties are stimulated. I will tell you a little bit about the genetic work. So this is the genetic structure of the maize panel we used. So if you are not familiar, this is a principal component analysis. We use 1 million markers to identify groups. So we have 5 groups here represented by key founders. In blue, they are the stiff stalk with B73 key founder. This is the first genotype from US being sequenced. And then OH43 and PH207 in the iodine, they are used to produce the commercial hybrids. So for the trial in France, we have Mogio here, 21 and 22. This is the grain yield being represented, but 22 was a really strong at the level of the temperature, so really high temperature and growth. You see here the performance in the stress during 2001 is above the control we had in 22. And the stiff stalk, they are outperforming, they are always producing more yield. And during very severe stress, the iodines are producing more. I will go first. This is genome-wide association we employ to identify genes potentially that can be used as markers. So we identify two genes described in fluorine. So the practical findings, so we used activities in laboratory and the field to determine the functional plasticity of crops to optimize the soil resource. So we've seen a considerable degree of variety. A large root system can sustain great shoot biomass. And at the moment, we are still developing markers for some desired traits. In the future, we will conduct functional studies to understand how these candidate genes or markers they can contribute to stress tolerance, and hopefully, so these markers can be incorporated in breeding program. So in the guidelines, we had to provide a question for you. So this is my question. So what are the potential long-term ecological impact of enhancing resilience in C4 crops? These are the different partners and PIs, and also a very big thanks to all the funding agencies which made this project possible. Voilà.
Good, thank you very much. We have time for one question. If you have more questions, yes.
Because there are so many markers already developed. Why do we need to develop markers?
Why do we need
New markers?
Yes.
Because what the old ones, are they not working, or not right, or but what is the argument? So to actually also to continue such work, such future work, or also to develop new markers.
So yes, there are some, but here the idea was to compare these two crops in parallel for the synteny to see which were the most relevant markers. And also we not only yield, so of course there are many markers for yield, but here we measured also different parameters using drones, so at the physiological level, and was to have a complete map of the situation. Roots also, so roots is considered as a black box in breeding programs, and it's quite recent that they are integrated and you have no markers for roots. And at this time, so we compare the root and the shoot at the same time to see if we can, we use laboratory and then pots and then field to see how predictive this can be. So if you do a screening in the lab, maybe you can already predict the performance of the root system in the field. It was to have I agree that there are some marks, but he loves to have a more complete picture.
Okay, but you can discuss that during the lunch if you want. I will continue because I want you also to have a good lunch break. And we continue now with the project BioHortiTech. So let's take this one. Okay, how do we get to full presentation mode? Does anybody know? In this on the right? This one? Up. Okay, very good. And the next speaker is Magdalena Scher. Okay. Okay.
Thank you very much. I will skip first slide because it's general, and I would like to save time to show you the results. So I will start from the objectives of our BioHortiTech project. So first objective was to find innovative formulations for biocontrol agents, microorganisms. We concentrated on Trichoderma fungi. And also we try to develop formulation for natural substances, and as natural substances we use essential oils produced by herbal plants. The next objective was to implement living mulching plant in production of horticultural crop, I mean in orchards, and also in vegetable production, field vegetable production. Next objective was combine both of those methods to check how they interact with each other, and it is really very important. We will see the results. We also, in this project, we also try to find how the microbial agents develop after application, because it is also very important to get more stable results using biological product in integrated and also in organic production. So first, this innovative formulation. So we prepared two kinds of formulation. First kind was all were microcapsules, because they are very convenient to use. So first kind of this formulation were microcapsules, lignin cellulosic microcapsules made of organic wastes to encapsulate spores of Trichoderma fungi. Another kind of capsules were capsules producing hybrid capsules, organic inorganic, which contained essential oils. So it was not very easy to produce such kind of capsule, because not all oils can be encapsulated and can produce stable capsules. However, we found several solutions, and next step was to examine these capsules in greenhouse in more control conditions, but we also concentrated on field experiments, because field is the best exam for all treatments. So the capsules we examined in cultivation of tomato plants. So you can see the experimental setup. How this picture? Why this picture? Because it was integrated tomato cultivation, but to check the effect of the capsules, we did not provide chemical control. So late blight killed our plants very quickly. Next, so we conducted those experiments within 2 years. So first year was complete disaster. However, we could save marketable yield, and the next year was much better, and we get very nice results. So what about those capsules? You can see that when we used the micro lignin cellulosic capsules, we got better marketable yield than in control without any treatments, and even better than in when we used commercial preparation Trianum. Compared to spores of Trichoderma, this effect of microcapsulation was much better. And also when we see the treatments, this right picture, when we see the microcapsules with essential oil, we found that essential oils, but not single oils, only mixture of the oils, were more effective in tomato cultivation than not encapsulated or single oils. So you can see this bar This is mixture of the oil encapsulated. So we can see that not only the total yield, but also the marketable yield was higher than in control. Of course, the increase was not very big compared to chemical control. However, it was really successful in field condition. So next experiment was connected to living mulching plants. So experiments were conducted in 2 sites, in Poland and in France, in organic orchards. And what was living mulch? Living mulch, there are plants which can be planted in rows or between rows instead of bar cover, because farmers usually they kill the plants in the rows in orchard by herbicides, or they keep the natural cover, but it is not very effective for plant growth. So what we expect from these living mulches, because they can do some services to crop plants. First, increasing the biodiversity in the ecosystem. Then we expect that they will provide some nutrients to the crop plants. Another thing is that they should attract pollinators, because they are we use some herbal plants, flowering plants, so they should attract pollinators. At the same time, they should repel pests. They should control weeds, and also they should have some positive impact on soil communities, microfauna, mesofauna, and microorganisms. What happened in reality, we will see. So this is the slide, how it looked like. This is control treatment. This is row of the peach trees with cover plants, with living mulch. And there were a lot of living mulches studied and selected during those experiments. However, we had to select the most efficient plant. What we got? There are some general findings. First of all, in young orchards, the effect of these living mulches was greater than in mature orchards, which were more stabilized. And in young orchards, unfortunately, the effect of these living mulches, opposite to expectation, was negative, because the trees, the diameter of tree trunk was reduced. Why? Because of competition, living mulching plant with young plants, competition to root development, competition for nutrients. Of course, plants used as living mulches reduce weeds and increased biodiversity in these rows of those plants, but not on the trees. However, it was found that the pests, the presence of the pests on the crop of the fruit trees was lower, but the differences, it was some tendency, the differences were not significant. Also, there were changes in nematodes population in the soil. However, there were a lot of variation depending on the kind of the orchard, age of the orchard, and also cover plants species. Living mulches increase microbial diversity, but only when it was estimated by new sequences, new generation sequences. When we use another methods, we didn't have any differences. And we found also that some special herbal plants have positive impact on the plants, on the trees, and other plants had negative impact. So the selection of the plants is very important. Next, we use the living mulches.
To come to an end.
Okay. Okay. So sorry, only short. We use also the living mulches, the intercropping plants in tomato production. And what happened? We use two herbal plants, aromatic plants, thymus and basil. In both years of the field experiments, thymus increased the yield of the tomatoes, basil decreased. Opposite to literature where the basil is recommended as a neighbor plants for tomato. And how it looked, how it worked. The tomatoes had greater fruit number and flower number, and also it increased development of root system compared to basil, which decreased compared to control. The also important thing is that in this experiment we applied also this bioinocula. And what happened? Bioinocula in integrated production and in organic production work good. But when we combined with herbal plants, these bioinoculations affect worse the plant growth and yielding. So combining of both of those treatments was not good for plants. And another last time it was also very important how the microorganisms applied to the different part of the plants can develop their population and what is the fate of this microorganism. So it was found that the roots, when the microorganisms are applied to the roots, they are more stable and they work better. But if we apply the microorganism to the upper plants, upper part of the plants, they work. Okay, sorry.
Sorry, but we have to
Okay, so if you have any question, please ask later. Thank you very much. And this is our team. Thank you, and sorry.
The good thing is you're here, so people have the chance to talk to you and to ask questions. So, Hervé, are you done with the presentation? No. Okay, so our last speaker in the session before lunch is Hervé van der Schuven. And so he will talk about the catch BNI crops. Okay, here you go.
Thank you for joining this presentation. I'm actually leading the project Catch BNI, which, as I explained in the pitch, is aiming at trying to reduce nitrogen losses in the agro system. So it's a kind of a relatively applied research, I would say, with different partners, as you will see. I will acknowledge during the presentation and at the end of the presentation. I realized from the other presentation I had to give that had too many slides, so I will skip a few, I can imagine, as I could see from the other presentation that it's always challenging to fit within 10, 15 minutes. I don't need to go back to these elements. You know the importance of nitrogen. We have estimated 70 billion euros, cost of 70 billion euros in terms of nitrogen losses in Europe. So you can imagine it's a very big problem. And there are so-called nitrogen directives that the EU is trying to implement in order to improve our management of nitrogen. And as part of this strategy, we want to introduce the concept that people are using for quite some time now, so-called catch crops. So those crops are supposed to take up any residual nitrogen in the agrosystems at the end after harvest. So we try to keep the soil covered with some crops that are able to take up this nitrogen and potentially to provide it to the next crop once it's incorporated in the field. It's an interesting concept that has been implemented quite successfully, I think, in the European agrosystems. But it's also interesting to think whether those crops, besides the fact that they take up the nitrogen, whether they can have some extra ecosystem services by basically bringing some functions into the soil. And so we divided our project in 4 work packages. I will mostly focus on work package 1, 3, and 4. The idea is first to check whether those cultivars, those species of catch crop do have a capacity to inhibit nitrification and denitrification. So for that purpose, we developed an assay, a bioassay, which is using an ammonium oxidizer, the nitrosomonas bioassay that we have, you can see here. And the idea here is to look whether the plants are able to excrete molecules in their roots in order to stabilize this nitrogen, so to stabilize the ammonium and prevent or slow down the conversion of ammonium into nitrates. So that's one assay that we use, and we use another assay which is more looking now at nitrification, denitrification, combining those two, looking at N2O emission, which is a greenhouse gas, as you might know. In this case, now we can have something a bit more holistic, let's say, because we look at how much when we incubate soil and bacteria with those tissues or those roots, how much N2O is being emitted. So we directly measure the N2O. We collaborate with a company that is actually developing so-called cover crops, catch crop. They have not looked into this perspective. It's a new angle for them. So they don't select or they don't breed for this purpose. They breed for something that grows fast and well, that farmers can establish in the field. But of course, you can imagine that if we could have some extra traits in those crops, that would be quite interesting. So what you see here is actually 2 results from the 2 bioassays. One is actually collecting root exudates from those catch crops. We call them CC1 to CC6. You see here, and we see within each catch crop, we selected the best performing cultivars according to the criteria of this company and how farmers are buying those cultivars. You see here, there's quite some variation in catch crop 1, for example. You see that if you take those 5 cultivars, you see that in terms of nitrification inhibition, we see that some can reach up to 60% of inhibition and others are going down to 20%. So there is genetic variation. It's a good news because every time there is genetic variation, as we have seen in other presentations, it gives hope that human beings are able maybe to select those traits or to improve those traits in the future for agriculture. We see that other genotypes perform less good, but still with some interesting inhibition, as you can see here. If we take the fresh tissue, that's another interesting concept that you have in mind. It's not only about the roots, how it performs in the soil, but since you grow those catch crop, eventually you are also able to incorporate those catch crop into the soil. So potentially the biomass, the canopy that is produced by those crops, they can also have a function or could have potentially a function, and we also want to measure that. So what we do here, we take the canopy, the leaves, we mix together with the soil, and we check whether we have reduction in N2O emission. What you see here, again, quite consistently, that this catch crop 1 is actually quite good because the emission is quite reduced as compared to other cultivars and species that we have been testing. So it's a kind of very promising, at least under control conditions. We have some good performance at the root level, but also at the canopy level. I will skip the work package 2 for the sake of moving further into the different concept, because work package 2 is actually, let's say, control condition testing of the concept, but you will see the results more in the field. It's also interesting to think, again, I'm bringing an additional concept. You know the issue of bringing organic fertilizer. The issue of organic fertilizer is we don't control very well the mineralization, nitrification, denitrification of those organic fertilizers. That's the reason why we control when farmers are allowed to bring manure to the field, for example, because we know it can have a peak of N2O emission, a peak of nitrate production, and of course, leaching into the soil. So there's also a very big challenge of stabilizing fertilizer, making sure that this fertilizer doesn't very quickly escape into the environment because the plant is unable to uptake all those nutrient elements. So in this case, what we want to do here is to see whether we have a potential to stabilize organic fertilizer with those catch crop. So the concept is to think, can we harvest those catch crop and help to stabilize the organic fertilizer? I should also mention that we have here the DMPP, which is a molecule, synthetic molecule has been developed by chemical companies that have a very strong capacity to inhibit nitrification. So it's a good control for us to compare to. It is highly debated, although there is some places, some countries where you can start to use, or we're using the stabilizer, the synthetic stabilizer. But of course, if you could replace the synthetic with something more natural, of course, that would also be a big advantage. You see here the results are quite interesting. You see that you can never compete with synthetic stabilizers, the DMPP, as you can see here on the graph. It's always outperforming any other approach. But you see here that if you mix a rhizospheric soil with those organic fertilizer, you are also able to reduce significantly. You need to compare the control, which is a non-treated, with a different catch crop. I should also mention that P44 is actually Persia 44, which is a wheat genotype, an ancient wheat genotype that has also demonstrated capacity to stabilize or to reduce nitrification. So there is quite some hope there, not to be as performing as synthetic stabilizers, but we can see that biological nitrification inhibition is something that has some promises. So, of course, there will be some need to optimize. What I'm showing you here is a kind of preliminary results, pioneering results. We haven't checked now different concentration, different combinations that could also help to stabilize those fertilizer. But I think going in the direction of trying to stabilize fertilizer is a good strategy for the future. We also tested this concept of catch BNI under field condition. Now I'm going back to the catch crop concept, so having catch crop on the field. And we test here, you see different genotypes that have been, or different species that have been selected, depending a bit on the place where we were testing this, in Germany and also in Spain. And we look a bit at what is happening in the field after incorporating this catch crop into the field. So we see that the different species have different impacts on the different on the biology, or let's say on the cycle of nitrogen. We also see, as you can see at the bottom part of this graph here, that the impact on ammonium oxidizer, being archaea or being bacteria, is relatively limited. So we don't see a very strong effect. If you would do this with synthetic inhibitors, you would have something quite strong in terms of inhibition. So again, minor effect of those catch crop in the field, as we can see here. We also see that depending on the catch crop, we can also look at what is happening to the cash crop, so the crop that is grown after the catch crop. In this case, it's wheat. And we see that in some cases, those catch crop are also able to stimulate or to improve the shoot elongation in the main crop. We see also that there is limited variation in terms, so it's very difficult to connect now to the end cycle because it takes into account many different elements, as you can imagine. So to link directly to simply the catch crop is very difficult, but at least we see that in our field trials here at IPK, we don't see major impact of having the catch crop or not having the catch crop. Having the following or having a catch crop, you don't see major impact on the yield, let's say, of the wheat, which is a good news. But of course, we would hope to see something, some kind of improvement of having those catch crop in the long term. So we've done also similar tests actually in different places. The John Innes Center has done the same kind of trials. Again, we don't see major differences in terms of nitrogen use efficiency, in terms of wheat yields. So it means all those catch crop, basically they have no negative or positive impact on the main crop that you have afterwards. We still need to see, I mean, that's a very difficult topic for us to research on, is how we can optimize nitrogen fertilization. You have to think about the terms that how this fertilization was organized. The idea is also to see whether with those catch crop, we can actually reduce the level of synthetic fertilizer in those fields. If you are able to better control nitrification, in theory, you should also be able to reduce the amount or the dosage of nitrogen that you provide to the field. So that's another kind of research that we are still exploring now. And I see that I will have to close soon. Again, here, what I want to show maybe as a last slide is looking at the impact. We have not tested only actually conventional catch crop, but we also started to look into some interesting ones like sorghum, which is another interesting catch crop because it has been shown actually by screening different genotypes from tropical areas. We've seen that some of those genotypes have quite strong BNI activity, so biological nitrification inhibition including sorghum. So this is work done by collaborators in the Basque Country. So they have tried different, or at least 2 varieties of sorghum to see their impact on winter wheat growth. What we see here is that one of the sorghum variety has demonstrated good potential. I will not go into the details, but we can meet later if you have more questions about those results. We see that it has quite some good capacity to stabilize, or at least to reduce the ammonium oxidizer population in the soil. We also see that it can also impact the different forms of nitrogen, while basically stabilizing ammonium in the soil when we use such a variety, in this case, the Vilomen variety in sorghum. We see some impact on N2O emission. Something you have to keep in mind, there's another interesting result, but it was known for quite some time. But we also have to keep in mind that when you incorporate those so-called green manure, so it means when you bring this tissue into the soil, of course, there is also mineralization of those plant material, this organic material, which is also participating in some kind of N2O emission. And that's also something we see now from all those different catch crops. So that's another concept that we need to keep in mind, is this extra organic matter that you bring. Although you want to target the BNI activity, you also have, of course, an issue with the organic matter that you bring in and the stabilization of this organic matter. I will skip this. We've done different tests of people in Basque Country have also done different tests with tomato. We also look at the quality of the produce, which is another important element to look at. We don't see a major change in the quality. So the idea is also to push the use of catch crop by farmers by also demonstrating that it has no effect or no negative effect, let's say, on the produce in the field. As I said, we are continuing. It's not finished. The project will be completed next year, 2025, early 2025. We're still ongoing experiments. We are bringing all those results together. We have some kind of multi-location trials. Now, as you could see, we had something in UK, in Germany, in the Basque Country, and of course, all results in more controlled environment. So we're trying to bring all these elements together to bring this as a publication. We believe, based on the result we have seen here, that there is a potential to use this BNI. We see variation in those catch crop. I think the role now is, well, there are 2 things, 2 main messages I could give you. One is, how can we genetically exploit this? So is there a way to further improve plants to accumulate more BNI capacity? And the other one is, of course, trying to bring those traits to the farmers with those improved varieties, and to see whether we can have an optimizing the use of those catch crop. We can see that depending on the timing of incorporation in soil, depending on the temperature, the climate conditions, all those important parameters are very influential, let's say, on the outcome of such a research. I think I will close it here. I need to thank all the collaborators. As I mentioned before, people at IPK, at the Basque Country University, at the John Innes, people at ULB in Belgium, people from my team in Jean Blou, but also, I don't see the sign there, but it's also companies and cooperative, ANCOP and DSV from Germany, providing very good genetic material for our research.
Well, thanks a lot for giving 2 presentations in 1 session, in this room and in the other room. Do we want 1 quick question, or are we going?
Have you observed any impact of the catch crops for the diseases,
soil-borne disease,
or any disease?
It's interesting that you're asking this. We tried to check. We actually just wrote last year a project looking into this direction with some team in France, Lyon. We haven't got it funded, but that's something definitely we have in mind. It's also to look at soil-borne disease using the focus here was very much on BNI, but you're right that these are also very important elements to look into. We believe that, for example, with cruciferous, it has, well, you know, the glucosinolates are included there. I think they can have some kind of antibacterial activities. So it might also be that such catch crop could be actually beneficial in terms of soil-borne disease. But it's a very interesting axis that we want to push as well, connected with research. Yes.
For soil stabilization, for soil colonization.
Yes.
Yes.
Okay, the people Online, do you have any question? I didn't check. Is there anything in the chat? There was something. Yes, we will most likely start a little bit later, most likely 1:45. So we have an hour for lunch now. Okay, so thank you everybody here, everybody online, and we see you in an hour.